Scientists tried to know whether all plants have phosphoglyceric acid as the first product of carbon dioxide fixation, or whether any other product was formed in other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of carbon dioxide fixation was an organic acid, which is the oxaloacetic acid. Since then, carbon dioxide assimilation during photosynthesis was said to be of two main types: those plants in which the first product of carbon dioxide fixation is a C3 acid, that is PGA, which is called the C3 pathway, and those in which the first product was a C4 acid, that is oxaloacetic acid, so the pathway is called the C4 pathway. These two groups of plants showed other associated characteristics also. Plants that are adapted to dry tropical regions have the C4 pathway. Those plants have C4 oxaloacetic acid as the first carbon dioxide fixation product. They use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. The C4 pathway is also called Hatch-Slack pathway. Is generally observed in monocot plants which grow in the tropical regions, and the C4 pathway is very clearly observed in the leaves of sugarcane. C4 plants are special. They have a special type of leaf anatomy. They can tolerate higher temperatures and they show a response to high light intensities. They lack a process called photorespiration and they have greater productivity of biomass. The C4 pathway operates in two kinds of photosynthetic cells, the mesophyll cells and bundle sheath cells. The bundle sheath cells are arranged surrounding the vascular bundles. Such an arrangement is called Kranz anatomy. The chloroplasts in mesophyll cells exhibit grana organization. The chloroplasts in the bundle sheath cells do not show grana organization, but they do possess thylakoids, but they are not arranged into grana. These two kinds of cells perform two different types of reactions. The light reaction takes place in mesophyll cells, while dark reaction occurs in bundle sheath cells. The arrangement prevents the oxygen evolved during light reaction from entering the bundle sheath cells. Thereby, there is no possibility of photorespiration. In C4 pathway, atmospheric carbon dioxide first diffuses into the mesophyll, and this carbon dioxide reacts with the 3-carbon substance which is called the phosphoenol pyruvic acid and forms a 4-carbon compound which is oxaloacetic acid. So as the first product, oxaloacetic acid is a 4-carbon molecule. This path is called C4 pathway. The primary carbon dioxide acceptor is a 3-carbon molecule phosphoenol pyruvate and is present in the mesophyll cells. The enzyme responsible for this fixation is phosphoenol pyruvate carboxylase or PEPcase. It is important to register that the mesophyll cells lack Rubisco enzyme. The C4 acid oxaloacetic acid is formed in the mesophyll cell. It then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release carbon dioxide and a 3-carbon molecule. This 3-carbon molecule is transported back to the mesophyll, where it is converted to phosphoenol pyruvate again, thus completing the cycle. The carbon dioxide released in the bundle sheath cells enters the C3 or the Calvin cycle, which is a common pathway to all plants. The bundle sheath cells are rich in the enzyme rubisco but lack PEPKase. Thus, the basic pathway that results in the formation of the sugars is the Calvin pathway, is common to C3 and C4 plants. Calvin pathway occurs in all the mesophyll cells of the C3 plants. In the C4 plants, it does not take place in the mesophyll cells but does take place in the bundle sheath cells. In the biosynthetic phase of photosynthesis, the atmospheric carbon dioxide combines with the ribulose biphosphate and forms 2 molecules of PGA. This carboxylation reaction is catalyzed by the enzyme ribulose biphosphate carboxylase or rubisco. This rubisco is the most abundant enzyme in the world, and it is characterized by the fact that its active site can bind to both carbon dioxide and oxygen. So it can act as an oxygenase as well as carboxylase. Photorespiration is a process of respiration which takes place in chloroplast in presence of light. In photorespiration, ribulose biphosphate is oxygenated in presence of oxygen, forming 1 molecule of phosphoglycolate, which is a 2-carbon compound, and 1 molecule of PGA, which is 3-carbon compound. The PGA molecule is used in the Calvin cycle. Phosphoglycolate is dephosphorylated, forming glycolate. This glycolate diffuses out of the chloroplast and enters into the peroxisome. In the peroxisome, it is oxidized and becomes glyoxylate. This glyoxylate is used in the synthesis of glycine. Glycine diffuses out of the peroxisome and enters the mitochondria. Here, 2 molecules of the 2-carbon glycine unite and form 1 molecule of serine, which is a 3-carbon compound. During this process, 1 molecule of carbon dioxide is released. Serine then diffuses out of the mitochondria and re-enters into the peroxisome. After some chemical reactions, serine is converted into glycerate. Glycerate diffuses out of peroxisome and enters the chloroplast. So here the glycerate is phosphorylated and forming phosphoglyceric acid, which enters the Calvin cycle. One molecule of carbon dioxide which was released in the mitochondria during the formation of serine will have to be refixed. So 75% of carbon lost due to oxygenation of ribulose biphosphate is recovered, and 25% is wasted as one molecule of carbon dioxide. Under the condition of intense light and poor availability of carbon dioxide, photorespiration plays a defensive role. With insufficient availability of carbon dioxide, the light energy cannot be fully utilized, and thus excess light can cause photooxidation and damage the plant. Here photorespiration provides protection against this. In C3 plants, some oxygen does bind to rubisco, and hence carbon dioxide fixation is decreased. In C4 plants, photorespiration does not occur. This is because they have a mechanism which increases the concentration of carbon dioxide at the enzyme site. This ensures rubisco functions as maximum carboxylase and minimum the oxygenase activity. This is the reason the productivity and yield are better in C4 plants.